It is a great pleasure to introduce to you Gertrud Schüpbach, Professor Schüpbach, from the University of Princeton. She's a member of the National Academy of Sciences, also the American Academy of Arts and Sciences. She's also a Howard Hughes Medical Institute investigator, a special professor, and then also she served also on the European Molecular Biology Organization, which is a very well-known and famous committee in Europe. She has been serving as a member of advisory boards and editorial committees, but I don't list them, holds many titles, medals, and awards. She is also very proud of the honorary degree that she received from the University of Zurich something like 4 years ago. Let me tell you that her scientific career started in Zurich back in the 1950s. This is a picture that was taken at the roof of the department in Zurich. This is Trudy, this is Eric, who is sitting over there. And then this wonderful gentleman used to be heading the lab. His name is Rolf Neutiger. And then, what do I do? Then in 1981, she moved to Princeton University, and this is the lab where she and also Eric works. Her interest was focusing on Drosophila eggs. This is a Drosophila female. She is busy in laying an egg, and once the egg is out, you realize immediately this is already a well-differentiated structure with an anterior end, with a posterior end, a dorsal side, and also a ventral side. The question how that polarization of the egg comes about is really an interesting one. It sounds like a sort of a silly idea to work on a system that leads to formation of an egg within those little tubes in the ovary of the female, but as you will soon see, that created lots of exciting things. Now, one approach to understand the way, the genetic basis of egg differentiation is simple induced mutations. And if you induce such a mutation, make a female homozygous with that mutation, then they will lay eggs which are not normal, but has got, let's say, 2 ventral sides, as this one is here, and of course this female is sterile. And in a set of mutagenesis experiments, she and then other groups isolated a bunch of such babies, such eggs, and as you see, this reminds you a gherkin, and possibly also a torpedo. So they named those mutations, and of course the corresponding genes, gherkin and torpedo. The other thing I need to tell you is that they are happy parents of 3 daughters. So they are also happy grandparents of 3 boys, which means that doing high-quality science and having a high-quality personal life is absolutely possible. The Wischhausen-Schüpbach couple is a wonderful indication of that. With this, I ask Trudy to come and take over the stage.
It's a real pleasure for me to be here as well and be able to tell you a little bit about the science that is happening in the lab. I enjoyed meeting some of you over the past 2 days, and so now I can actually get to give a presentation here. Some of you have given a presentation to us, so it's nice to speak to you again. And Janusz gave a very nice introduction into the problem that we are studying in the lab, but I will sort of repeat some of the things that he said in more details. So my laboratory has been interested in this question of how in development do you actually establish asymmetries? Why, if you look at, say, here an adult fly, but you could ask the same question about the human being, about the frog, about the worm. Why is it that we start with an egg, which is a single cell, a fertilized nucleus is inside, and then this develops eventually into an organism that has a head at one end, a tail, in this case, at the other end, has a dorsal side. In the fly, the dorsal side has the wings, the ventral side has the legs. How's this organized? If we start out with one nucleus that has all the DNA that is present in the genome and then divides into cells, and then somehow these cells will have to know, okay, we are here at the anterior of the egg, and we have to form a head. We are here, other cells will end up here, they have to form the tail, and other cells have to know that they're on the dorsal or ventral side. So if you look at this Drosophila egg, as Janos pointed out already, we can predict by looking at the egg, because the egg is already asymmetric. We can already predict these major axes. We will know that this is anterior, this is posterior, dorsal and ventral. We can just look at the egg because it's oblong, it's longer along the anterior-posterior dimension. It has specialized structures here at the anterior, and on its dorsal side, there are these protrusions. We call them dorsal appendages, so we know here is the dorsal side, here is the ventral side. So then the question in my lab was, well, so how does this then happen? How do you make an asymmetric egg, and what's inside the egg that then allows an organism to have just one head and a tail and all of this? Well, the answer, of course, did not come from our lab alone, but many laboratories were studying this problem, and it became apparent that during development of the eggs, there are maternal RNAs that are placed into the egg by the mother. So, for instance, here is a Drosophila egg at an earlier stage, but you can see here highlighted a particular RNA that corresponds to a gene that's called Oscar, and it is deposited here at the posterior pole. Very, very specifically, this particular RNA, although it's made by the mother in the so-called nurse cells, it's transported through the egg and then is anchored here at the posterior. Such localized RNAs are not only specific to Drosophila, you find them in many eggs of many different animals. Here is, for instance, a frog oocyte. It's studied in the laboratory of Doug Melton, Harvard, and you can see here in white highlighted another localized RNA. It's called VEG1. It is localized to the vegetal pole of the egg during oogenesis by the mother. These localized RNAs then become translated in the embryo into protein by the translation machinery, and these proteins often form gradients. So here is another example from the fly. We have the so-called bicoid RNA, which is deposited by the mother at the anterior end. After the egg is fertilized, it's translated into protein, and you can see there's a lot of this protein at the anterior end, and then there is a gradient where there is not so much here, and then there is nothing at the posterior pole. And the cells that are the embryonic cells in this egg can read and respond to the level of this protein, so that anterior head genes are activated in this region of the embryo. Thoracic, which is in the fly, sort of the middle of the body, genes are activated here. And then there is another protein gradient that results from this original Oscar RNA. There is another RNA called Manos RNA, which is deposited at the posterior pole and makes now a gradient from the posterior. And this gradient activates posterior genes, abdominal genes. So now we understand something about development. We have these eggs. The mother puts RNAs in different parts of the egg, and then they are translated and are acting to activate specific gene transcription in the embryonic cells in different parts of the egg. So then my lab was wondering, well then how come that some RNAs go to the anterior, some go to the posterior, and then some things which I didn't show are activated on the ventral side versus the dorsal side. So in order to understand this, we have to go back in oogenesis, because as I said, when the egg is laid, these major axes are already there. So in order to understand how this comes about, we have to look at oogenesis. Here you see a Drosophila ovary, and as János has pointed out to you, these ovaries consist of these strings of egg chambers. These egg chambers derive from stem cells which divide at the tip of the ovary, and then these egg chambers mature, they grow in size, and at the end then the egg is laid by the mother. Now if we look at such an egg chamber in more detail, we can actually also already predict the future anterior, posterior, and dorsoventral axis. Here is the oocyte. This is the cell that will become the egg. And at this stage of development, the oocyte is connected at its anterior end to a set of nerve cells. These cells are very large nuclei. You can see one of them here. And they are very synthetically active. They make RNA and protein, which is then transported through canals into the oocyte. So there is an anterior-posterior asymmetry here already built into this egg chamber, because the oocyte is at what's considered the posterior end of this egg chamber. Here will be the posterior pole of the egg and embryo, and here will be the anterior end of the egg and embryo. And the dorsoventral axis we can also predict, because the oocyte nucleus is a little hard in this slide probably for you to see, but here is a blue blob, which is the oocyte nucleus stained with a blue dye, and it sits very asymmetrically within this oocyte. You can see it's very close to the edge here and far away from this end, and wherever it is, that is going to be the future dorsal side of the egg and embryo. So as the egg is being built by the mother, there are already asymmetries. And what we have learned over time, as I will explain, is that these asymmetries are made during oogenesis in simple manners, and then they are signaled to the overlying cells, the follicle cells, by the oocyte, and then the follicle cells send signals back to the oocyte, which then inform the RNAs that are being produced by the nerve cells of where they have to go and where they should not go. So in order to get at this problem, we chose to do genetics, so do a genetic analysis, and induce, as János pointed out, use a mutagen, which we fed 2 males in this case, mated these males, bred the generations, and created homozygous mutant females of unknown mutations, and then tested these females to look whether they would lay abnormal eggs that would give rise to abnormal embryos, telling us that the genes that we had mutated would be normally required to make the wild-type egg and embryo. So here, I'm showing you a couple of examples of such mutant eggs that we isolated. Again, here's the wild-type egg, and you can predict, for instance, the dorsoventral direction of the embryonic development by looking at these dorsal appendages, which are on the dorsal side. And you can see that in this egg here, there are no dorsal appendages, and in fact, this egg is what we call ventralized. It only has ventral structures, and the embryos that develop inside these eggs also only make ventral structures, only make ventral mesoderm, which is a ventral structure in insects. In contrast, here is an egg that we call dorsalized. Here, we have, instead of just 2 very narrow defined populations of cells that make these dorsal appendages, here the cells, the follicle cells from all around the egg were thinking they were dorsal. They made these huge dorsal appendages, and the embryos inside, if they form, they only make dorsal skin. They're dorsalized as well. So these mutants, therefore, gave us a handle on genes that are normally required in the mother, because we had homozygous females, right, making the abnormal eggs. So that tells us that these genes are required in the mother, the wild-type genes normally, to make a normal egg that has a normal polarity. So then, of course, one is always very interested what do these genes encode? And doing molecular work, we found that these mutations here were in genes that in fact had well-studied human homologs. So the gene, as Janos was telling you, where the names come from, this torpedo gene turned out to encode a homolog, a fly homolog of the vertebrate EGF receptor. And this gene here, the mutation in this gene here, which we had called gherkin, turned out to encode a TGF-alpha-like signaling factor. And from vertebrate studies, it was already known that this transforming growth factor alpha is a ligand. It can bind to this EGF receptor. So what is this EGF receptor? Well, it's well studied in humans and in human tissue cultures because it is, as the name implies, it's a receptor that can receive signals. So there are signaling molecules, and these are homologs. These signaling molecules can bind to the receptor and activate its enzymatic activity, are also conserved from flies to human. And here's, for instance, gherkin that is made by the oocyte and can bind to the receptor and activate its activity. Here is a picture I took from some review on cancer, because the EGF receptor is one of the very important genes that is often mutated in cancers, in various cancers, and there are a number of drugs that have been developed specifically against the activity of this receptor. So in this tissue culture cell, in this scheme, you can see the receptor sitting at the membrane. It can bind these growth factors, and if it binds the growth factors, it becomes active. It activates a downstream cascade of genes which do a number of different things. In particular, they promote in tissue culture cells proliferation of the cells. They block apoptosis. If you have a mutation in this receptor that makes this receptor overly active, therefore you have over-proliferation, which you can imagine is not good, of tissue. You start forming tumors. If there are more mutations in the pathway, it can lead to metastasis, invasion. The cancer cells become immortalized because apoptosis is suppressed. So it's a well-studied receptor in this context. However, in normal cells, of course, it has not only proliferation functions, it has other functions as well. And so in the fly, it's involved in pattern formation during oogenesis. Here, this is a little hard to see, I guess, because of the light here. Unfortunately, you have to imagine this is an egg chamber, and the receptor is expressed, shown in white here, in the follicle cells surrounding what is an oocyte here. And then down here, you have to imagine again that if you look very closely, there is actually an oocyte here that is about this size here. There are the nerve cells over here. And in this oocyte, you see the gherkin protein very localized within the oocyte. So here we have this theme again of the mother making RNA or protein and localizing it within the cell, this time around the oocyte nucleus. And this is a signaling molecule, so it will be released from the oocyte, it will travel over to the follicle cells, which are not so visible here, which sit around the oocyte and will induce dorsal cell fates in these overlying oocytes, follicle cells. You can maybe see it better here. Again, here is the egg chamber, here is the localized RNA, and then here, after the RNA is being translated, you can see the protein, now shown with an antibody against the protein here, sitting in a very localized manner within the oocyte. And it will now signal to these overlying follicle cells and induce them to be dorsal by activating dorsal-specific genes within these follicle cells. And then these dorsal-specific genes are not activated on this side, and therefore these cells develop as ventral follicle cells. And then at a later stage, these follicle cells send signals back to the oocyte, which then instructs the embryo about where the dorsal versus the ventral side should be of the embryo. Now, the question then, of course, becomes also, it's always in science, you find out one thing that's exciting, like a localized RNA, and that establishes dorsoventral polarity, and then immediately you have 10 other questions about this. So you're always going on and on, you're never finished in a way with your projects, because now the question is, why is the RNA here? How does the RNA even know that it should be here and not just everywhere within this oocyte? Well, so we were looking at some of the other mutants that we had isolated, and we realized that the mutations that dorsalize the egg and embryo are involved in the localization process. So again, here is a wild type. You can see the gherkin RNA very localized here above the oocyte nucleus, or in a cross-section. You can see it here. Here's the nucleus, here's the RNA. And in these mutants, the RNA is not localized just to this corner, but it's found in a ring all around the oocyte. So localization has failed here. We have mislocalization of the RNA, and therefore what happens is in this mutant case, we get now protein made all over the oocyte here, and now all the follicle cells receive this signal and they activate their EGF receptor, and they activate the downstream genes, the dorsal genes, which normally should only be active on the dorsal side, but here, because now we have protein all over, they also become active on the ventral side, and therefore we get these very abnormal dorsalized eggs and embryos out. This particular mutant is called squid because it sometimes has like multiple dorsal appendages and looks like a little squid that swims around. One of the pleasures of working with flies is that we give our mutants often very funny names, and it's easier sometimes to remember the genes if they have a funny name than just some number or some strange abbreviation. So looking now over all of these dorsalizing mutants and doing experiments, also biochemical experiments, finding that some of them bind to each other and that some of them in fact bind to the gherkin RNA, we came up with the following model where squid and another gene that doesn't have such a nice name, HRB27C, bind to the gherkin RNA in the nucleus as it's being transcribed, and they leave the nucleus with the RNA, and then in the cytoplasm, they recruit other binding proteins to the RNA. These proteins that are now bound to the RNA have two functions. For one, they are translational repressors, because you don't want the RNA to be translated before it has reached its destination, because you want the protein to be only in one place within the cell. So the RNA, as it comes out of the nucleus, has to be transported, and some of these proteins associated with the localization machinery motor the RNA along microtubules to its place of destination, close to the oocyte nucleus, where it is then anchored and starts being translated into protein after it's been localized. So this is our scheme. As I said, we have used some biochemistry to show who binds to who and who binds the RNA, and also done some live imaging to test about the transport of this protein complex within the oocyte. However, the remaining 5 minutes or so, I'd like to actually go on, because studying this RNA localization over time, we became aware that this RNA is already expressed earlier in oogenesis. So here is a smaller egg chamber, and you can already see the gherkin RNA being filling up this small oocyte. So over the time of oogenesis, the whole thing grows, and especially the oocyte gets bigger and bigger over time. So here it's still very small, but the gherkin RNA, and in fact the protein, are already in there. And we became aware, studying now more carefully earlier oogenesis, that the gherkin protein, which you can see in this very small egg chamber here, is already signaling to the follicle cells at this stage of development. But in this stage of development, it induces posterior follicle cell fate in these posterior follicle cells. What happens in response to this, the follicle cells just here at the posterior take on a posterior fate. That means they start expressing posterior specific genes, and then they signal back to the oocyte, and that signal that they send back to the oocyte has the function that it changes the microtubule cytoskeleton inside the oocyte, and these microtubules drive the oocyte nucleus away from the posterior end and into a corner of the oocyte, and wherever it lands, it's sort of pushed away, and wherever it lands at the anterior, that's going to be now the future dorsal side of the egg and embryo. And you can see the gherkin protein here, it's still signaling posterior. Now the nucleus has moved, and it starts accumulating now on one side to much higher levels than on the other side. And so this now establishes a dorsoventral asymmetry. With other words, this signal and receptor are used twice in development. First, to establish an anterior-posterior polarity, and then later, the dorsoventral polarity. Maybe you can see, here it's a little bigger, so maybe you can see it better. Here is the early signal. Gherkin is in the small oocyte. It signals to these posterior follicle cells and makes them posterior. And then they signal back, the oocyte nucleus moves, it's here now in the corner, and now the gherkin protein, RNA and protein accumulate on one side, and now it signals and makes these follicle cells dorsal. As a cartoon here, you can see, this just in cartoon form to repeat, we have the first time this signaling happens, makes these follicle cells posterior. They signal back. The oocyte nucleus moves to one corner, and we have now the new signaling through the same signaling molecule and the same receptor, but now these follicle cells will become dorsal. So then, again, new questions arise. What is that green signal here that the follicle cells make? And we're still working on this, so I don't really have an answer right now. Another question is, why is there a different response to this signal at the two different time points? You get posterior cell fates at this time, and then the same signal and same receptor at a later point induce these cells to be dorsal. So what's the difference between this stage and this stage? In order to get at this, we again made did screens. This time we screened not homozygous females, but we screened directly in the egg chambers for mutants in which if we had a mutation at the posterior end in the follicle cells, and I won't go into all the technical details here, but these cells here are, for instance, mutants. The black cells are mutants, the green cells are wild type. So here we have an egg chamber where all the follicle cells are mutant. Mutations in which the signal from the follicle cells to the oocyte would fail, and so the oocyte nucleus stays at the posterior, and with it the Gherkin protein stays at the posterior. At the time in development when in wild type the oocyte nucleus should be up here and the Gherkin protein should be up here, and now you can see here the nucleus is still here and the Gherkin protein hasn't moved, which is the indication that this second signal has failed to be produced by the follicle cells that should establish the anterior-posterior polarity of the oocyte. The interesting thing, after we cloned some of these mutants, we found that some of them block Notch signaling. Notch is another signaling pathway where one cell, in our case the oocyte, sends a signal. This signaling molecule is called delta. It's received by another receptor in the follicle cells called Notch. And again, there are downstream genes that are activated when this Notch signal is active and in fact enters the nucleus and regulates transcription in the nucleus. So what this indicates is that the difference between the early anterior-posterior signal is that we need, in fact, two signals. We need both a signal through the EGF receptor and a signal through this Notch pathway. So there are two coincidence signals in a way that both produce transcriptional changes in the nucleus, and if you have both of them, you become a posterior follicle cell. If you, at the later stage, have only EGF receptor activation, you become a dorsal cell. And so this is how you initially establish these differences that are then carried forward in development. So again, if you look at the egg chamber, now we know that initially we have a signal here at the posterior. I have two signals here that come together through Notch and EGF receptor. So we make the posterior here, and then here on the dorsal side later, we only have one signal, EGF receptor activation, that makes you dorsal. So obviously this is the beginning when you establish these broad axes in the egg, which then help to localize these maternal RNAs that are then present in the egg only at the anterior, only at the posterior, or only active on the ventral side. And then these maternal RNAs, of course, that the protein made from these maternal RNAs activate the whole other programs, like a head program or a tail program, and so on, so that eventually over time, you end up with an organism that has just one head at the right end and one tail, and in insects' case, 6 legs, 2 wings, and everything in the right place. And with that, I will end. I also have to thank many people over time in the lab. I couldn't list even all of them. I just list the major students and postdoctoral fellows. Many of these names are actually graduate students in the lab who have been working on these problems. I have to thank the funding sources, and last but not least, the FLY and the FLY community, who produce a lot of useful tools and have a lot of input always in our work. And thank you for your attention.